Okay.
Good morning. I'm Cynthia Magro. I'm the Director of Skin Pathology for Weill Cornell Medicine. Dr. Annetta Rezko asked me to speak for a few minutes on some very interesting research that we've done on COVID-19. So what I'm going to review for you this morning is really the pathology and the pathophysiology of a very unique subset of COVID-19 infection referred to as severe COVID-19. Now you have to understand that the vast majority of people who are infected with COVID-19 have either no symptoms or relatively mild disease. So what I'm about to describe is a very minor subset of patients, but obviously the morbidity and mortality in this particular group is quite high. And certainly when you listen to all the and watch all the various media presentations, it does tend to focus on this severe COVID-19 subset. So, what exactly happens in these patients that develop severe respiratory distress syndrome and other, multi-organ failure? So that's going to be kind of the focus of my brief talk, and it's going to be in the context of our paper where we outline the pathophysiology of severe COVID-19. So the paper that I'm referring to is this one that has been published in Translational Research entitled Complement-Associated Microvascular Injury and Thrombosis in the Pathogenesis of Severe COVID-19 Infection. There were a number of collaborators, and they're all listed. Myself, Justin Mulvey, David Berlin, Jerry Noovo, Steve Salvatore, his resident, and Dr. Jeffrey Lawrence. So let me go over the findings that we made in this interesting paper. So what I'm showing right here is basically normal appearing lung. And I'd like to focus in particular on this slide right over here. Basically what one is, and over here, what one is looking at over here are the alveolar spaces. The alveolar spaces are very key because they actually contain the oxygen that is critical for distribution to the rest of the body. Now, how does the oxygen from the alveolar space get to the rest of the body? That is, obviously a very important and critical question. It does so through the septal capillaries. And so what we see over here are these delicate thin septa. And these thin septa contain blood vessels that are referred to as the septal capillaries. So again, to adequately exchange oxygen to the rest of the body, the breathed in air ends up in the alveolar space, and that oxygenated air then transfers through the septal capillaries through the left ventricle to the rest of the body. So what happens in severe COVID-19 infection where these patients develop acute respiratory distress syndrome? And I'm going to be talking a few minutes on the pathology, the pathophysiology of the lung injury that we think is going on in these patients. So this happens to be a collage of images of a damaged lung in a patient who died from severe COVID-19 infection. I would say that this case and another case that I'm going to show you really defines the prototypic pathology that we see in severe COVID. So here, the alveolar spaces, instead of being clear, they're actually filled with all these red structures. Well, these red structures are red blood cells. So there's a lot of blood in the alveolar spaces. And so why does that happen? Well, as it turns out, the septa that I showed you in this slide, where they're very thin and delicate and contain these healthy blood vessels, the septa in severe COVID-19 infection capillaries that have been profoundly injured. And I'll go through the mechanism of injury. So if we look over here, the septa actually are destroyed. The capillaries are damaged. They have fibrin deposition in the wall and in the lumens. And as a consequence of this capillary injury, there is capillary compromise, the release of red blood cells into the septa and into the alveolar spaces. And so you can imagine that with such damaged capillaries, that the normal pathway of oxygenation in a lung that is not diseased is completely interrupted, and therefore the natural transfer of oxygen into the capillaries in severe COVID-19 infection doesn't occur, and as a consequence the patients really become profoundly hypoxemic. So when you look at this pattern of injury, to me it is very intriguing. I would define it as a very destructive capillary injury pattern. It's somewhat inflammatory, but it's not strikingly so. There are no viral alterations, like no viral cytopathic changes, which we see in certain viral infections like varicella and measles. And in fact, the pattern of injury, to my eye, is one that resembles complement-mediated microvascular injury. And so with that in mind, we actually studied these cases for evidence of activation of the complement pathway to sort of prove that somehow this virus activates the complement pathway in the septal capillaries, destroying them and interfering with oxygenation. So this is just a diagram of the complement pathways that are operational in our body. We all have them. They're part of the innate immune system, and while they're very important in dealing with infections and other exogenous antigens, the pathways can also cause a lot of tissue destruction if they are overzealous, and that's probably what is operational in severe COVID. So we have three main pathways: the classic, the lectin pathway, and the alternative pathway. And when these pathways are activated, they will ultimately generate a molecule down here, which we call C5b-9, the membranolytic attack complex. And that molecule, that C5b-9 structure, actually injures the cellular target. So when you activate any of these three pathways, the end result is the production of C5b-9, which in the setting of the COVID lung would damage the lining cells of the capillary, which is the endothelium. So this dysregulated overzealous complement activation within the septal capillaries damages the endothelium through C5b-9. At least that was our espousal. So we were able to more or less prove it in our studies. We were able to show that components of complement activation, such as C4d, C3d, and C5b-9, were present in the septal capillaries of the lung, indicative that this capillary injury, in fact, was mediated by complement, and that is what is illustrated here. These delicate septa, the capillaries, a lot of complement, and then again, when you activate the complement pathway, you will produce that MAC, which injures the cell. This is another lung, and it shows the same pattern of injury. You have these air spaces that contain red blood cells, and look at the septa. The septa are damaged. The capillaries contain fibrin in their walls and in their lumens, referred to as blood clots. It's not too inflammatory, which is typical for complement-mediated vascular injury. And once again, we can see the complement components being deposited within the microvasculature of the lung. C4d Again, the effector mechanism of that vascular injury, which is C5b-9, is very extensive. MASP2 is indicative of activation of the mannan-binding lectin pathway, which we think is operational in severe COVID, and it too is positive. Now, severe COVID is not just a process that involves only the lung. Actually, it's a systemic condition, and extrapulmonary injury can occur, including in the brain, in the heart, in the kidney, and in the skin. And in fact, the skin is a window to severe COVID-19 infection. These patients can develop a very distinct type of skin rash referred to as retiform, which is net-like purpura. And here we have an example of a patient. This is his buttock showing this very striking purpuric retiform rash. And just like we saw in the lung, where we saw all that fibrin deposition in the capillaries, again, complement mediated through some mechanism, in the skin we see the same pattern of injury. The blood vessel contains a large clot, a fibrin thrombus. And once again, just as we saw in the lung, where you have all that complement deposition, which is key in mediating the injury, as well in the skin, we have a lot of C5b-9 in the microvasculature. This is another patient from our series. Again, these are all severely ill patients that have respiratory distress syndrome through the mechanisms that I outlined. Here we have a patient with acral levato, so the net-like purplish rash on the foot. Clots are found in the artery, and again, it's a complement-mediated process with a lot of C5b-9. Now, interestingly, if you biopsy a severe COVID patient, if you biopsy their normal skin, specifically the deltoid skin, you can prove systemic complement activation by demonstrating in totally normal-looking skin the MAC, or the C5b-9, within the microvasculature. And so in this image, we have totally normal skin that contains the MAC within small blood vessels of the normal skin. And this again is another patient. Note the net-like skin rash. What we're illustrating here are tiny little thrombi in venules, and again it is complement mediated, so we see the C5b-9 within blood vessels, and because it is a systemic process, when you biopsy normal skin, you will see C5b-9 in the blood vessels of normal skin, which is illustrated over here. Now, this is some interesting work that Dr. Nuovo, he's a very well-known pathologist who specializes in identifying viral proteins and viral RNA in tissue sections of pathology specimens. And he was actually able to demonstrate that the coronavirus spike glycoprotein is found within the septal capillary endothelium of the lung. It doesn't mean that the virus is actively replicating in the microvasculature, but at least the protein has somehow ended up in the endothelium. And so that's interesting when we try to sort of sort out mechanisms by which the complement activation occurs. And so what we see here are the septa that have all those capillaries, and he's used a software technology where the C4D, which is the complement, is fluorescing green. He then has an immunohistochemical stain for the spike glycoprotein of coronavirus, which again, using his nuanced software, is highlighted in red over here. And then when he sort of superimposes the images, you get a yellow signal. And you can see how the capillaries, which have all that complement, also show spike glycoprotein as revealed by the yellow signal that indicates that you have both complement and the spike glycoprotein colocalized, which we know has to be in the context of the microvasculature of the lung. And this is in the skin, actually, of one of those patients who have that retiform purpura, where we were able to, once again, as we did with the lung, show the colocalization of the complement with the spike glycoprotein. And again, it doesn't necessarily mean that the virus is actively replicating in the endothelium, but at least microparticles are in the endothelium and may have a role in the complement activation. Now, as you know, to actually diagnose coronavirus infection, there is a specific sample that's used, which is the nasopharyngeal swab, and that's because the virus will have a predilection to infect certain cell types by its ability through the spike glycoprotein to bind to ACE2 receptors. So this is courtesy Dr. Nuovo, where we have this nasopharyngeal swab. It's being stained with H&E, that stands for hematoxylin and eosin. And you can see that the glandular cells of the nasopharynx, they don't look terribly healthy. They've lost their cilia, that's referred to as cilia cytophthoria. And here you can see some of these degenerated glandular cells because they've been infected by the virus, since the nasopharyngeal cell does have that ACE2 receptor to bind the coronavirus. So the actual point of entry of the virus, whether you have mild coronavirus infection, you're asymptomatic, or you have severe COVID, which is something that happens in 1% of people infected, the point of entry is the nasopharyngeal area. And this just shows you using both an in situ RNA technique as well as immunohistochemistry, we can actually highlight the coronavirus SARS-2 RNA in these degenerating nasopharyngeal epithelial cells. It has a brown signal. So this is actually from our paper, and it sort of outlines the pathophysiology of the severe COVID-19 that I've tried to show you the pathological images. And basically, I'd like to go over this diagram because I think it's very instructive. So number one is we have the virus, and this is the virus, and it's called coronavirus because there's a crown of glycoproteins, of these spike glycoproteins, which we see right over here. And you see these spikes will bind to the ACE2 receptor, and there are only certain cell types that have that ACE2 receptor, and they encompass the nasopharyngeal cell, cells in the lung that we think are at least macrophages and pneumocytes. Dr. Nuovo thinks that there could be active infection in the endothelium, but that's debatable. But do the endothelial cells really have the ACE2 receptor? But anyway, the ACE2 is found certainly in the lung in macrophages and pneumocytes, that we know. It's found in intestinal epithelial cells, so there for some patients with COVID-19 infection actually have diarrhea. And we also find it in the brain. So what happens when the coronavirus is incorporated into the cells that have these ACE2 receptors, the cells are destroyed. The angiotensin converting enzyme 2 is destroyed. So we get a plummeting systemically in the angiotensin converting enzyme 2 levels. So it really becomes this systemic process associated with this reduction in ACE2, which in the severe COVID-19 patients is obviously quite profound. Now, this ACE2 reduction is really important in terms of understanding the severity of severe COVID-19 infection and how it can affect different organ systems. When you have this reduction in ACE2, because the ACE2 has been destroyed by the coronavirus, you end up with an imbalance in the angiotensin 2, angiotensin 1,7 system, whereby the ACE2 reduction leads to an increase in angiotensin 2, and that is not good. Angiotensin 2 actually results in endothelial cell injury, and that is probably a critical mechanism of endothelial cell injury and potentially of complement activation. There is one study that shows a high level of angiotensin II systemically is associated with complement activation. So that is, one mechanism by which the complement pathway, which we know damages the endothelium, could be activated. Another potential way is the coronavirus itself. Because it has those, glycoproteins, those glycoproteins can bind to the mannan-binding lectin pathway. There are certain sites on the mannan-binding lectin pathway that will bind the glycoprotein from the coronavirus, and that is another mechanism of this complement activation, is through the direct interaction of the mannan-binding lectin pathway with the glycoprotein of the coronavirus. And that is, again, another mechanism of complement activation. And again, we produce the MAC, which is what damages the endothelium. When you damage endothelium, or through MAC, or this angiotensin II system where you have the production of NOx2, the result also is vascular thrombosis. So when you damage endothelium, either through complement activation or more specifically through the effects of high levels of angiotensin II, you activate the clotting pathway, and that will result in vascular thrombosis, which could potentially be widespread. And so the implications in terms of therapy in patients who have severe COVID-19 infection would be utilizing drugs that are anticoagulant in nature because of the procoagulant sequelae of this severe form of COVID-19 infection. Complement inhibition therapy also may play a role, definitely, in severe COVID-19 patients. And another interesting sequelae of the angiotensin II associated endothelial cell injury is a high level of IL-6, interleukin 6. And interleukin 6 has actually been shown to have procoagulant effects. It actually causes platelet aggregation. So a very interesting line of therapy in the severe COVID-19 patients is anti-IL-6 therapy. Now, the question is, why is it that only a very, very small percentage of patients actually develop this form of COVID-19? Well, it's fortunate that only a very small percentage of patients develop severe COVID-19, but it's probably a number of factors. Certainly, obesity is a huge factor. The fat is an inflammatory organ, and likely there is significant complement activation in the fat, so we know that that is certainly a risk factor and ties in very nicely with the severe COVID-19 pathogenesis. The other is diabetes mellitus. Diabetes can be associated with complement dysregulation. Why? Because the regulator of C5b-9, or MAC, that molecule that's generated through complement activation that sort of drills the hole into the endothelial cell, the regulator of that molecule of C5b-9 is in fact CD59. And in diabetics, the glycosylation of CD59 renders it inactive. So these patients already have a reason to have an overzealous complement pathway. Obviously, if you have pre-existing lung disease, that is certainly a risk factor. And then we have to look at the perfectly healthy patient, the exceedingly rare scenario where somebody who is perfectly healthy, not obese, but they develop the severe COVID-19. And there it's likely genetic factors. Maybe they may have some undetected complement protein gene abnormality that could be operational, similar to what is implicated in atypical hemolytic uremic syndrome. So I'd like to close by saying, we know a lot about the pathophysiology of the virus. It's a very clever and sophisticated virus. I think that the vast majority of people who are infected with COVID-19 have a self-limited illness, including 25% of patients who are asymptomatic. The severe COVID-19 is a fascinating entity, but it, of course, it's very serious, and it is very uncommon, but that's what we hear a lot about. and that is really the role of this virus in this very unusual population subset that I have described: obesity, diabetes, maybe some complement regulatory protein gene abnormality, where we have this systemic process going on with complement activation and vascular thrombosis, with the main targeted organs being certainly the lung, the skin, brain, and kidney. And I think that is more or less all I have to say. I hope you were able to understand some of it, or even most of it. And our paper is available online that you can always read. Thank you.